So in the interest of time, I'll go straight on to the ELN guidelines. So the European Leukemia Net, despite its name, isn't really a specifically European organization. It's funded by the European Union, but there are representatives not only from Europe, but also from, I think, Korea, Australia, and the United States. So it's really a global collaboration, although it originated in Europe. So this is a group of experts who get together on a regular basis to discuss new developments in CML, to review the results of clinical trials, and to try to incorporate all of the new research into an easy-to-understand set of guidelines for use by CML clinicians. So the current version of the ELN guidelines were published last year in Blood, and that was the third version of the guidelines. So the ELN guidelines, if you haven't read them, they contain a definition of chronic phase, accelerated phase, and blast crisis. They produce a very nice summary of recent clinical data on CML. So it's kind of a one-stop, this is everything that's important that's happened in CML in the last few years. And then they set these response definitions, which, like the comment from before, is a bit like a red light, yellow light, green light definition. So optimal means that this person is on track to have a normal life expectancy. So this person is unlikely to die from his or her CML, and therefore there's no reason to change treatment. The worst group, the failure group, this person is doing so badly on treatment that they should change to another drug to prevent the development of accelerated phase or blast crisis or to prevent death. And then in between is a kind of a gray zone where they're not doing so badly that they should change drugs straight away, but you should be keeping a very close eye on this patient so that if things continue to get worse, you can institute a change before it's too late. So they're not doing so well that you can be completely relaxed, but they're not doing so badly that you should do something about it right now. So they've changed some of their response definitions. So it used to be that they had a warning group, which basically said that this person is someone you should watch closely. They've now done away with the, they used to have a suboptimal group, which was the gray zone, and they've now changed it to the warning group. So instead of saying this person is somewhere in between, they're saying now that you have to watch this person very closely. They have changed the monitoring definitions, so that there's a greater reliance now on qPCR and less of a reliance on cytogenetics. And they've changed the core treatment recommendations. So the major changes are that it used to say that everyone got imatinib first and then switched to another drug later should the need arise. Now it says that any of the first three drugs, imatinib, nilotinib, or dasatinib, is a reasonable first choice. And in second line, they've added in the use of bosutinib or ponatinib. So the study then goes on to put people into these different categories at different time points. So this is newly diagnosed patients. So if you have a high-risk SoCal score or other risk score, or if you have additional chromosomal abnormalities in addition to the Philadelphia chromosome, then this puts you into a slightly higher risk group that should be watched more closely because we'd be less relaxed about how you're going to go. After 3 months, we come back and do a PCR test or chromosome test, and we then divide you into one of these risk groups. So if you have a BCR-ABL of less than 10% at 3 months, you're on track to have a very good long-term response, and that's good. If you're more than 10% on any drug, then this is considered a warning. So this is considered an indication to keep a close eye on how this person is going, to evaluate regularly, maybe to look for mutations if things are not going well. If you haven't achieved a hematological response, if you still have a high white cell count and a big spleen at 3 months, then you have to change drug because that's very, very poor. And so what you see is that this is 2009 on the left and this is 2013 on the right. So effectively what's happened is that the suboptimal group from 2009 is now considered a failure. And the optimal group from 2009 has now been split into still optimal or the warning group. So we've made the requirement more stringent. We're trying harder than we were 4 years ago. So here again, this is the 6-month time point. So you've made it through your 3-month time point, and now you're coming up to your 6-month result. So here again is 2009 on the left and 2013 on the right. So if you've now dropped to less than 1% by 6 months, or achieved a complete cytogenetic response, which is approximately less than 1%, then you're doing very well and you're on track to do well in the longer term. If you've dropped below 10% but not below 1%, then you're in this warning group where you might do okay, but things might go badly, so you should be keeping a close eye on things. And if you're still more than 10% at 6 months, then this really is failure of treatment, and you should be switching if that option is available to you. And again, if we compare 2009 to 2013, we've moved the suboptimal group into failure, and we've split the optimal group into a really optimal and a not quite so good warning group. And then again at 12 months, so this is after 1 year of treatment, the ELN 2013 now says that a major molecular response, so less than 0.1% at 12 months, is an optimal result. If you've got between 1% and 0.1%, then you're in this warning group where you still might do okay, but it's not completely relaxed. And if you haven't achieved a complete cytogenetic response, or you're more than 1% at 12 months, then this would now be considered a failure and a reason to switch. There used to be ELN guidelines for 18 months, but they've now been dropped. So again, this is emphasizing this. It's what you do early that matters. It's getting a good response quickly is very important. At any time point after 12 months, a continuing stable or improving molecular response is optimal. It's a warning if you are in this in-between CCR and MMR group, and it's failure if you lose any of these previously achieved responses. In this little warning group here is something that we haven't talked about before, and this is people who develop cytogenetic abnormalities that are not in the CML. So these are people whose CML is in remission, but they've developed new abnormalities in the bone marrow. And so there have been rare cases of patients developing acute leukemia or myelodysplasia in the non-CML population. It seems to be very rare, but there are a number of cases reported from around the world. So if you happen to pop up one of these things, then that's also an indication to keep a closer eye on things. So going back to that curve I showed you an hour or more ago, at 3 months you should be down to here, down to 10% or less. At 6 months you should be down to 1% or less. At 12 months you should be down to 0.1% or less. And after 12 months you should be anywhere below that line, and that would be an optimal response on any drug. I might skip over the second line recommendations because I think we must be running out of time, and it's probably more important to have time for questions. If you make it to the third line, you should still be having a transplant even now, because we're not confident that any of the TKIs will give you a good long-term response, so there is still a case for transplantation. Pat mentioned that there's also a set of NCCN guidelines from the United States. These have traditionally been more conservative than the ELN, and I think that globally probably more people follow ELN than NCCN. Interestingly, the NCCN has just recently adopted more molecular criteria, so it is coming closer to the ELN, but there are still some differences. I don't think I have time to go through, but they are less strict than the ELN criteria. So in the last slide, I've tried to predict if the ELN produces a new set of guidelines in 2015 or 2016, what questions will they be looking at? One question is accelerated phase. Is there a better way to define accelerated phase than the things that we've been using for the last 20 or 30 years? So this is something that the ELN considered last time, but they couldn't agree on. So this question might come up again. Is there a better way? Accelerated phase was defined in people treated with interferon, hydroxyurea, and so on. Should we be having a different definition of accelerated phase for TKIs? But we haven't been able to agree on that. The ELN avoided having more than 10% as a failure, and I think that that was an area in which the consensus group disagreed. So there were some people who felt more strongly about the 10% group. So is there some way that we can look at those very early time points to get a better separation of those who might be rescued later on and those who might be on track to do badly? As we collect more data about people who... So most of our data are for second-line treatment are people who started imatinib and then switched to nilotinib or dasatinib. In many countries, lots of patients are starting with nilotinib or dasatinib and then switching. And for that group of patients, we don't actually have a lot of data to guide what to do next. So hopefully there will be more data on those questions in the near future, so we might get better guidance from the ELN next time. The other question is, what about deep molecular responses and the chance to come off treatment, so-called treatment-free remission? Is that something that could legitimately be considered a goal of treatment for some or all CML patients? I think there's no universal agreement on that. I think there are some people who are very enthusiastic and think that it's very important, and there are others who feel that really the most important thing is to be alive with a good quality of life, and that anything beyond that is the cream on top. So that's something that we may see some refinement of in the future. And that's the end of my talk.
You've been so brilliant. It's time actually to get out for tea, but
Vijay,
we'll take
can I have
5 minutes
one
at
short
least.
comment?
Yeah, just a second. Let me finish what I'm saying. Yeah.
All right, okay, sorry.
We have time for at least a couple of questions, for sure. I'll start off with one small question. We are constantly telling patients to be compliant. All the time compliance is, the patient's responsibility. But I'm listening to you on the ELN guidelines. How compliant are physicians in following these guidelines?
Oh, I don't think anyone's ever dared to look. But I think that the answer is probably not great. Probably not great, I suspect. I don't know. I think in Australia, most people pretty much follow them. But Australia is a relatively small country in population terms. And because Tim Hughes has had such a major role, I think he has influenced the way that CML is treated in Australia. So maybe that means that there's more compliance. But I suspect globally there's enormous variation.
Especially in this region, we know for sure.
Yeah.
Not because of it's deliberate, but because of the given conditions. Yeah.
And I was pleased to hear that there are sort of patient-friendly versions of the guidelines, because if your doctor is not following these guidelines, then you should be asking.
Absolutely. Yeah. So just a couple of questions. Yeah.
I have heard that in the US, 75% of patients do not get monitored as per the guidelines in the United States.
Also, I comment, Doctor. On the data for CML-related deaths because of serious side effects, I have a concern in the methodology of the studies, even the use of the phrase untimely death because of CML. You see, in rural areas where death certificates are being done hastily and unresponsibly, it doesn't exactly say in the death certificate the cause of death is CML. More probably, it is the latest disease at the time of the death. This may corrupt the database for the studies.
Well, I mean, everything that I've shown you has come from major international clinical trials. So these are all being undertaken in academic centers, and these patients are being monitored very closely. So this is a very controlled population where I think we have very good data on what the cause of death was. So I think because these are clinical trial data from academic centers, I don't think that's an issue. I think more broadly it might be an issue, but for these results, I think we can be fairly confident.
We have time for just one more question.
Okay, I have a question. Just in the EHA Congress in Milan, at the first education session of CML is about stem cell, leukemia stem cell, and interferon alpha.
Mm-hmm.
And last year there is a paper in leukemia about it's a review of emergence of interferon alpha. But it seems that some physicians have different opinions about the combination of interferon. alpha and TCAT. I wish to know about your opinion.
So look, I think that interferon is a very interesting drug. I mean, you saw from those curves that there are some people who did exceptionally well on interferon, and the first treatment-free remission was actually interferon, that there were people who'd been on interferon for years who stopped it, who remained in remission. The numbers were very, very small, but it was there. So I think interferon is a very interesting drug that potentially does still have a place. The problem is the toxicity is quite considerable. And one concern has been that if you try, as I said before, if you try to combine two different drugs, you have two lots of side effects. And I would hate to think that you gave somebody interferon and they reduced the dose of imatinib because of the side effects of interferon, so that you could potentially lose something by combining the two drugs. So I think you have to be very careful. In Australia, the ALLG study for newly diagnosed CML has just opened, the latest studies. We're now on to CML 11. And in CML 11, it's nilotinib and interferon. So we have enough confidence, or enough interest in it to be doing a clinical trial. But in that study, what we've done is we start nilotinib first to get good control in the first few months, and then we add in a low dose of interferon later, once we've already got the nilotinib sorted out. Because the previous studies that combined the two drugs started them both together, which means that you then have all these side effects all happening at the same time. So we've tried to separate that out to make it safer.
Thank you so much. What you've done is really Himalayan. A big round of applause for Dr. Ross, please.
Thank you.